Thank you very much. Hello, everyone. I'll just bring up my pointer. So, hi, my name is MoranFrenkelPinter, and I'm a new assistant professor at the Hebrew University in Jerusalem, Israel. Due to bandwidth issues, I will stop my video sharing, but I'm very happy to tell you, to present to you today work that I did at Georgia Tech as part of the Center for Chemical Evolution and the NASA Center for Origins of Life, where I worked with Alcomari Guzman Martinez from the University of Mayagüez from Puerto Rico, and Nick Hudd and Lauren Williams and Luke Liman and other great collaborators. And I'm very happy to share this today. And I would like to thank also the organizers for giving me the opportunity to present this work. So let's start. All right, so chemical evolution, we've heard a lot about this. I also, like Sarah, just want to point out, bring up this slide where we really, I think it will be great to really think about, different steps during chemical evolution and how different mechanisms of, information and memory could have been transferred during this era before we had the very sophisticated polymers of today, where we can have really Darwinian evolution. So how did memory evolve? How did polymers evolved and information and so on? And so we know about these different steps where we have different pool of monomers going to oligomers through covalent and non-covalent interactions. And we could think of course, a lot of heterogeneous polymers, but also some, more homogeneous ones due to different selection forces. And then throughout evolution, chemical evolution, we would expect to see, increasing structure and new functions, which come up from polymers that were selected for due to hydrolytic stability and so on. And we'll see things like homochirality and eventually, will lead to life and so on. Specifically, the talk today will be focused on this step from monomers to oligomers. So what could have been protopolymers that could have been really interesting in this transition, or to facilitate fast transition and robust polymerization. And we all know during chemical evolution, when we talk about this process, that when we look at meteorite composition and model prebiotic reactions, we know that all of today's, the building blocks of all of today's biopolymers, of nucleic acids, sugars, of proteins, so their building blocks are found in meteorites and in model prebiotic reactions. Specifically for today's talk, I will discuss what could have been, proto versions of proteins, which are now just chains of amino acids, but what could have been, these earlier forms that polymerize more robustly and could also have been functional and form structures. And when we talk about, formation of peptides and protopeptides on the prebiotic Earth, we encounter several obstacles. And the first one, which is of course very important, is the thermodynamic barrier. So to make a peptide bond or an amide bond between amino acids, this is what we call condensation dehydration reaction. So we need to lose a water molecule, and this is going to be unfavorable in aqueous solutions. But we also have entered a problem of a kinetic barrier. So to make the peptide bond, it requires this high activation energy. And another problem is that once we have a dipeptide like this one, it can cyclize into this diketopiperazine, which, following another dehydration, which is a side product that will be really hard to hydrolyze, and it's kind of a dead end in terms of if we think of longer polymers. So how can we overcome these obstacles? So for number one, so for the thermodynamics, so what we and others like to do is really do, for instance, a dry down. So if we have dry conditions, we are spontaneously we would form these products through dehydration. It will be favorable. But for the kinetic barrier and the cyclization, it's really to overcome this, we can, for instance, go to higher temperatures or use high energy molecules. But in the Center for Chemical Evolution, we found another really nice solution, which comes from expanding the repertoire of molecules that we're looking at, as opposed to just looking at today's biology. So as I mentioned, if we just take amino acids and we would dry them down spontaneously, we should form peptides or peptide bonds or amide bonds, but it just requires a lot of energy. And what in the Center for Chemical Evolution, Nick had suggested is really looking at hydroxy acids. So hydroxy acids have, instead of the amine group, they would have the alcohol group. And when we dry these down, and Irina Mamajano did these experiments, so when we dry them down, what we form really pretty easily, that means at lower temperatures, we form these polyesters. So instead of peptide bond, we have ester bond. And we find a lot of polyesters in biodegradable polymers, in our disposable cups, and so on. But this reaction is very easy, so at low temperatures. And what Ram Krishnamurthy suggested is really what happens if we'll take amino acids and hydroxy acids together. So it's true today we have in biology these chains of amino acids, but we know that hydroxy acids were very abundant on the prebiotic Earth, and they look, of course, very similar. So what happens if we put the two together? And the answer is that we get copolymers of hydroxy amino acids or of ester and peptide containing molecules, which are called deoxypeptides. And the reason why this works, so we form peptide bonds, but I just mentioned that it's really difficult to form a peptide bond. But this is really because when we form an ester, we now have an activated site where the amine can attack, and we get a peptide bond instead of an ester bond. So basically, we're getting to the same place, so a peptide bond, but we didn't need to climb a really tall hill to get there. We just climbed, I guess, through a much lower energy barrier to get to the same point, which is great. So we get deoxypeptides. When we looked at this, and we're very excited about these molecules, we encountered several, I guess, limitations. So while these are great molecules, we knew that this would work under acidic conditions. And we needed temperatures which are pretty mild, but we thought that when looking at this, we thought that maybe there's an even more robust way to do these, to form these peptide bonds. And this is coming from, a lot of work about the importance of thioesters and, of course, thiol-containing compounds. And this has come a lot, in works on prebiotic chemistry and the importance of thioesters from a lot of groups. And this is originally, I guess, was the thioester world was originally suggested by Deduve. And many researchers have been continue to work on this, where we know that, for instance, biochemistry uses a lot of thioester intermediates. And the question is, could these thioesters really, could they have drove chemical evolution through these intermediates in a similar manner if we look at a similar system? So this is what we're thinking. We're thinking that molecules that are will be called mercapto acids. So this would be thiol and a carboxylic acid containing molecule. We speculated that they will, if we take these mercapto acids, that if we dried these molecules, so under low water activity, we would form thioesters. And then similar to deoxypeptides, we thought that amino acids, if we dried amino acids together with mercapto acids, we can form these products of peptides that will also have thioesters, which will be called thiodeoxypeptides. So that was the hypothesis. And to test that, we came up with this experimental design where we tested several mercapto acids. This is, for instance, thioglycolic acid, which is the mercapto acid analog of glycine. We tested this thiolactic acid, which is the mercapto acid analog of alanine, and so on. And we tested several amino acids such as glycine, alanine, phenylalanine, and so on. And what is really important here is that, so we took, in our experiments, we usually use a single mercapto acid and a single amino acid, and we dried them down together, but under anaerobic conditions. So if we would do these, if we heat up these molecules under, in a normal atmosphere, we will see quite a lot of oxidation. So very importantly, we do it under anaerobic conditions, and then we form different oligomers, and we actually form a variety of oligomers. And the main challenge is characterizing what we got, which molecules we got, different compositional. I just heard something. Different compositions and so on. So we use a variety of analytical techniques to look at the composition and properties of molecules that were generated, including NMR, high performance liquid chromatography, mass spectrometry, and so on. And I'm going to show you some of the characterization that we did in order to really elucidate and understand what were the products of our reactions and how robust they are. So here I'm showing you what happened after we took thioglycolic acid and L-alanine, and we dried them down together at 65 degrees Celsius for 1 week under anaerobic conditions. And we're looking at the mass spectrometry, which shows us the intensity over M over Z, and we labeled some of the. So what we see here is really the composition of the different products. For instance, this could be one product that has 1 thioglycolic acid and 2 alanine. Here we'll have 4 thioglycolic acid and 2 alanines, and so on. So we form a variety of different products. And we also looked at the, basically, we used the chromophores on these molecules and wanted to see the distribution of different oligomers. And to do that, we separated the products using a C18 column, which separates based on hydrophobicity on an HPLC. And what we saw is really that we have a variety of different polymers, and this is coming from the lower chromatogram is from just taking the thioglycolic acid, where the upper one is if we introduce the amino acid into the mixture, we're going to get really these copolymers of these mercapto and mercapto and amino acids. And so we knew through FTIR analysis and our HPLC analysis, we knew that we formed peptide bonds. But another way to make sure that we also really have what we call thiodepsipeptides, which have both thioester and peptide bonds. We looked at their UV vis, so we could record during the separation of the molecules, we could really see their full, absorbance. And when we have a peptide, then what we'll have is an absorbance spectrum. So here we're looking at a peptide running our column. It's a 3D. So we look at absorbance over retention time, over wavelength from 190 to 300 nanometers. And so usually a peptide will have maximal absorbance at around 190 to 100 nanometers. But when we look at the thioester containing compound, the maximal absorbance would be at about 235 nanometers. So when we look at our compounds separated on our column, we can see this kind of shoulder showing us that they're also thio at 230 nanometers, which showed us that there are multiple products indeed in our mixture that contain thioesters. So we do form thiodepsipeptides, and we have a whole mixture. And very important part of our characterization was really to validate some of the products that we had in the mixture. And that was using synthetic standards that were synthesized by Dr. Luke Lehman from Scripps. So Luke synthesized a bunch of standards. And this is, here I'm showing just one of them. So we're looking at also another validation of our products from proton NMR. So basically we're just looking at this molecule right here that has, it's our thioglycolic acid linked with an amide to alanine. We verified that this was, and this is basically the upper panel is a non-reactive mixture of thioglycolic acid and alanine. This is a comparison to the standard synthesized by Luke. And this is after dry down for 1 week at the bottom panel C for 1 week at 65 degrees Celsius under anaerobic conditions. And this is just to show that part of the product, and actually a major part, is really this synthetic under this dimer. We wanted to explore the range of conditions in which these reactions work. And I mentioned that, for instance, with deoxypeptides, when we have hydroxy amino acids, we know that they would form mostly under acidic conditions. But we thought and speculated that these reactions actually with mercaptan acid would work at higher pH as well, and maybe we'll be able to see that. So we tested the reaction at different pH conditions from 3 to 7. And here we are I'm showing another C18 column separation. So basically each peak is a product, and we kind of label some of them, TGAs, 1 TG and 1 alanine, and so on. And we really see that even at neutral pH, pH 7, we do see some formation, but it's more robust under acidic conditions. Something else that we see is when we tested the temperatures at which these reactions work, from 25 degrees Celsius to 65. So far, I showed you the 65 degrees Celsius, but we do see that, of course, as we lower the temperature, we see less formation of products. So there are less peaks in the range where we'll have higher retention time, but there's still some formation. And I guess that it's okay for a chemical evolution to take some time, more than a week. So this definitely works, it just takes more time. So to summarize this work, what I've showed you is that we see that when we incorporate mercaptan acids into monoabiotic peptide reactions containing amino acids, we see that really mercaptan acids catalyze peptide bond formation through the thioester intermediates, and that these thiodeoxypeptides are formed over really a wide range of conditions. We have varying hydration levels. I didn't show this, but we also have some formation in water, not as much as a dry down, but still some thioesters. And then peptide formation is done in water. Varying temperature conditions and a broad range of pH conditions. And really, we think that incorporation of even simple mercaptan acids, as these ones into these systems, might offer new avenues for dynamic combinatorial chemistries in water or under dry down conditions. And of course, deoxypeptides could have formed in the periodic Earth and participate in early stages of chemical evolution. And with that, I would like to thank my collaborators on this project. So, of course, Professor Aiko Marie Guzman Martinez, Professor Nick Hudd, Professor Lauren Williams, Professor Luke Lehman, Professor Facundo Fernandez, Marcus Bauza for the mass spec analysis. I would also like to acknowledge and thank fruitful discussions with Professor Ram Krishnamurthy, Arthur Weber, and Dr. Anton Petrov, and the funding agencies, NASA Postdoctoral Program for funding me, the funding agencies from the Center for Chemical Evolution and the NASA Cool Center, and basically, of course, all of my lab mates throughout the years. And I also want to mention, as I mentioned, I'm a new assistant professor, and I'm looking for recruiting talented master students, PhD students, and postdocs. As I mentioned, I'm in Israel at the Hebrew University, just started. Great place to be at, very international, and I'm funded by the Israeli Early Faculty Program. And thank you for listening. I would be happy to take any questions. And this